Cut an apple in half, leave it on the counter for 10 minutes, and the bright white flesh you sliced into turns a dull brown. The same thing happens to a ripe avocado or a raw potato. Most of us look at that rapid discoloration as a sign of spoilage. However, the tissue isn't rotting and the cells aren't dying. That brown stain is the visible wreckage of a plant's chemical weapon, deployed the exact instant your knife ruptures its tissue. To understand the brown, we have to isolate the biological machinery driving this reaction inside the cell. From there, we can reverse engineer the chemistry behind the techniques used to disable it. Every time you prep produce, you are engaging in molecular warfare against a sophisticated set of plant defenses. Inside an intact plant cell, two reactive ingredients are kept in strict quarantine. An enzyme called polyphenol oxidase, or PPO, is stored in the chloroplasts, while defensive chemicals called polyphenols are locked inside the vacuole. When your knife ruptures these microscopic lipid bilayer membranes, the quarantine fails. The enzyme floods out, the polyphenols spill loose, and the ingredients mix. The reaction then requires a third variable to ignite. Ambient oxygen from the air rushes into the newly exposed cellular tissue. The PPO protein pulls in ambient oxygen and combines it with spilled polyphenols, outputting reactive, colorless o-quinones. These unstable molecules immediately undergo polymerization, linking into larger chains that absorb light and darken. This forms melanin, a sticky, toxic physical barrier that walls off damage and gums up invaders. Lacking a circulatory system or white blood cells, the fruit relies on this localized instant immune response to defend its wounds. Applying a squeeze of lemon juice to an apple launches a precise biochemical strike, neutralizing the reaction at the molecular level. This 3D model shows the PPO enzyme's catalytic engine, two central copper atoms. Ascorbic acid from lemon juice clamps onto these atoms, locking them down. Simultaneously, citric acid forces the environment from an optimal pH of 6.5 down to 2.8, causing the enzyme structure to completely collapse. This two-pronged attack shuts down the catalyst's engine while simultaneously crashing its optimal working environment. For foods like potatoes intended for frying, where acidic flavors are unwanted, you can target the enzyme's structural fragility instead. Enzymes rely entirely on maintaining a highly precise, carefully folded shape to remain functional. PPO functions best within a specific thermal band, peaking at temperatures between 35 and 50 degrees. Submerging the tissue in boiling water triggers thermal denaturation. As the temperature rises toward 100 degrees Celsius, the protein structure permanently unravels into a disorganized, non-functional strand. Blanching permanently destroys the molecular machinery, ensuring the reaction cannot run regardless of how much oxygen is present. Physical barriers, such as submerging peeled potatoes in water or covering guacamole with plastic wrap, offer a third way to win the battle. These methods work by starving the reaction of oxygen, the essential third substrate required to begin the cascade. The massive fresh-cut produce industry uses this exact same principle of oxygen deprivation to protect millions in raw inventory. Commercial shredding operations like the massive lettuce processing shown here must combat the enormous surface area of ruptured cells exposed to the air. Many facilities utilize refrigeration. While cold storage doesn't stop the enzyme, it reduces kinetic energy, dragging the reaction to a crawl. By manipulating the thermodynamic conditions and substrate availability, these engineered barriers effectively prevent the plant's natural chemical weapon from firing. Humanity has spent centuries battling the economic losses of fruit browning, a struggle visible in the historical scale of apple harvests. Modern agricultural science has produced a biological solution to this ancient problem, the Arctic apple. This engineered fruit carries a specific genetic modification that downregulates the production of the PPO enzyme. Without the enzyme present in the cell, mechanical damage allows oxygen and polyphenols to mix indefinitely without ever producing melanin. A fruit that refuses to turn brown provides the definitive proof that the PPO enzyme is the singular driver of this evolutionary defense. A simple brown spot on an apple slice is a high-speed chemical defense cascade that has been evolving for millions of years. Understanding the chemistry of copper chelation and thermal denaturation shifts the kitchen from a place of rote instruction to one of precise chemical management.